I would like to welcome you today to the second intermediate filament seminar of this season. Our speaker today is Professor Dr. Daniel Starr. He is an Associate Dean of Research and Professor of Molecular and Cellular Biology at UC Davis. His research focuses on the mechanisms that govern the movement and positioning of nuclei within cells. Defects in nuclear positioning can lead both to developmental defects and diseases such as lissencephaly and muscular dystrophy. His lab uses mainly C. elegans as a model organism to study the role of KASH and SUN proteins in nuclear cytoskeletal connections. Dr. Starr's work has revealed key insights into the nuclear envelope's molecular mechanics, including its role in cell development and migration. Dr. Starr is also involved in mentoring and leadership, directing the PrEP at UC Davis program. He was named an Allen Distinguished Investigator in 2020 for his contributions to cell and developmental biology. Today, we are very excited to have him give a talk on giant KASH proteins and how they maintain the biophysical properties of the cytoplasm in vivo. Daniel, the floor is yours.
Thank you to all the organizers and for that wonderful introduction and for having me here today. I'm really happy to be here. And today I'm going to tell you about how these giant KASH proteins maintain the biophysical properties of the cytoplasm in vivo. And we know from classical experiments that the entire cell is mechanically coupled. So in this classic experiment here where you have this tissue culture cell and they pulled on the edge of the tissue culture cell with a glass rod, you can see everything is connected to each other and there are the forces are being transferred from here at the edge of the cell to the nucleus is being deformed, and even the nucleoli are moving in the direction of the pulling. So everything is mechanically coupled, and it's led to this this model of force propagation through mechanical transduction, and that forces generated outside the cell can be transferred across the plasma membrane through integrins into the cytoskeleton, including intermediate filaments and actin filaments, and then across the nucleus through the link complex, which I'll talk a lot about today, into the nucleus to the structural elements of the nucleus, including the nucleus skeleton and chromatin. And that way you can propagate these forces on the millisecond time scale to forces from outside the cell can actually change gene expression inside the cell. And what I want to focus on today are the link complex here that transfers the forces from the cytoskeleton across the two membranes of the nuclear envelope and into the nucleus to the lamins and the chromatin. And so this is our model of how link complexes work, or this was our old model. I'm going to tell you there's some problems with this model throughout the talk today. And how the link complexes work is that the SUN proteins go to the internuclear membrane where they interact with lamins in the nucleus skeleton and then have a long coiled domain across the space. And then here, the SUN domains, they interact with these little 30 amino acid luminal domains called KASH peptides of these KASH proteins. And the KASH proteins are known as nesprins in mammalian cells. And the ones I want to focus on today are these giant KASH proteins, which are known as Nesprin 1 and Nesprin 2 in mammalian cells, ANC1 in C. elegans, and MSP300 in flies. And all of these proteins are giant, 800 to 1,000 amino acids. They consist mostly of spectrin repeats. And at the C terminus, they have this transmembrane domain and the KASH domain to get to the outer nuclear membrane. And at the N terminus, they have CH domains that bind actin. So we think this is a tether. It holds nuclei in place by attaching the nuclear envelope to the actin cytoskeleton. And link complexes now have been implicated in a lot of cell and developmental processes, of course, mechanical transduction, but also the origin of eukaryotes, the architecture of the nuclear envelope and the ER. I've been studying them primarily from the role in nuclear migration and anchorage, but they're involved in the development of many, many, many different tissues. And then not surprisingly, they're involved in a lot of diseases from progeria, sterility, muscular dystrophy. But really, I want us to think about these giant KASH proteins as being involved in neurological disorders, ataxia and ALS. So here, from a meta-review published a couple of years ago about clinical mutations associated with the SIGN1 gene that encodes giant Nesprin-1, most of the patients due to defects in Nesprin-1 giants are neurological disorders. So most of them are ataxias and then ALS, and then a few muscle diseases. So I want us to think of these giant KASH proteins as playing roles in neurological diseases. Okay, so back to our model, and these giant KASH proteins. In the old model, the thought was that they tether the actin cytoskeleton through this calponin homology actin binding domain to the outer nuclear membrane through this transmembrane domain KASH domains with interactive links. And then they have this giant stretch of mostly spectrin repeats that can be on the size of up to 1,000 amino acids. So these are giant proteins that are thought to tether one end, the nuclear envelope, the other end, actin. And I'll tell you throughout this talk that this model is probably way oversimplistic, and I'm going to develop a new role for what these giant KASH proteins might be doing to regulate the biophysical properties of the cell. So we've been interested in this giant KASH protein, ANK1, through its role in positioning nuclei, and as was mentioned in the introduction, primarily in C. elegans. And so this is a worm, a C. elegans worm, and there's a GFP in the nucleus of these of the skin cell, the hypodermis. And the hypodermis of the worm is a syncytium. So there's about 140 nuclei in this syncytium, all lit up here in GFP. And as you can see in wild type, these nuclei are very evenly spaced apart. And as the worm moves around and crawls, these nuclei will sort of move around a little bit, but they stay in their place and they stay evenly spaced apart. In an ANK1 mutation, so you get rid of this giant KASH protein, you can see the nuclei cluster. They're no longer anchored in place, and they can move back and forth as the worm moves around, and you get these clustered nuclear phenotype. And we can score this nuclear positioning defect by counting the percentage of nuclei that are touching each other and therefore unanchored. Okay. Now, in this model, the old model was that ANK1, the giant KASH protein, was anchored to the nuclear envelope through its interaction with SUN proteins. So in this model, the hypothesis would be if you deleted the SUN proteins, you should get this same phenotype. And we were, when we carefully measured this a few years ago, we were surprised to see that that wasn't what we got. So here are the same data I just showed you. The ANK1 mutant gives large numbers of nuclei clustered or touching each other. So on order of 50 to 60% of the nuclei are touching each other. This is a strong nuclear anchorage defect. The UNC84 mutant has a few nuclei unanchored, but you can see it's far less than ANK1. So these phenotypes are not the same. The ANK1 phenotype is much more severe, suggesting that ANK1 has roles in nuclear positioning that are independent of the link complex. And that's what I'm going to focus on mostly today, roles of ANK1 that are independent of the SUN proteins. And so then what is ANK1 doing? So here's another schematic of ANK1. This is the data I just showed you. ANK1 nulls have a large defect. UNC84 nulls have a fairly mild nuclear anchorage defect. And so if we get rid of just the KASH domain here, we see the same phenotype, suggesting that this is link independent, the difference from here to here. What about the calponin homology domain? That had no phenotype. So we were surprised to see that for nuclear positioning, you didn't need the actin binding domain. So what was working? We found out that the transmembrane domain was very important for this process. This is all published, so I'm going very fast. The transmembrane domain is important for this process, and large stretches of the spectrin repeats were important. So just to conclude from this published research, the CH domains are dispensable, and the link complex plays only a minor role in nuclear positioning, and this transmembrane domain is important, and so are these spectrin-like repeats. So we started to think, what would the transmembrane be doing that's independent of the rest of the link complex? And the thought was maybe it's at the ER. And while ANK1 is enriched at the nuclei, at the nuclear envelope of some tissues, in most tissues, it's not enriched at the nuclear envelope. And instead, in this CRISPR tag GFP ANK1, you can see ANK1 is spread throughout the entire ER of the worm. So here's an ER marker, and here's ANK1. You can see they co-localize. These large holes would be nuclei, and you can see it's not really particularly enriched at the nuclear envelope in this tissue, though it is in other tissues. So we shouldn't necessarily think of ANK1 and giant nesprins as a nuclear envelope protein as much as they are more ER proteins. So if it's an ER protein, we thought we should look at the ER. And so here's a real-time GFP ER marker, and this is in the syncytial hypodermis of the worm. And you can see the worm is sort of crawling around. And the large holes here are nuclei, and then the rest of this is the ER network, okay, in these cells. And you can see as the worm moves around, the ER sort of stretches and compacts a little bit, but it's moving as a unit, and it sort of all stays in place, all right? Now, when we introduce a null mutation in ANK1, we see severe defects in the ER. And so here is just removing this one gene. And now the ER is no longer anchored in place. And it's we call this phenotype sloshing. It's sloshing around in the cell. The ER has become fragmented. You can see the nuclei are moving back and forth in the flow here. And this is just a mess. So, So our conclusion, and we also looked at mitochondria in this paper a couple of years ago in eLife, mitochondria, lipid droplets, everything is sloshing around. We effectively turned this nice ordered cytoplasm where organelles were all in their places into a soup where the organelles are all sloshing around. And we've drastically changed the biophysical properties of these cells by getting rid of this one gene, ANK1. So then what might ANK1 be doing? We came up with a couple different hypotheses here that the ANK1 could somehow regulate constraint of the cytoplasm through scaffolding the ER or other organelles or cross-linking the cytoskeleton. Alternatively, ANK1 could be regulating the molecular crowding of the cytosol at the macromolecular level. Now, the problem here was I'm classically trained as a developmental geneticist, and these types of hypotheses, I just don't have the tool set to answer them. And this really required an interdisciplinary approach to tackle this problem at the interface of developmental genetics and biophysics. And in order to do that, I needed a collaborator. So I was very lucky to recruit Gant Luxton to join me at Davis as a co-PI. And so all the work I'm going to show you today, Gant is, this is an equal collaboration between Gant and I running a joint lab that we now call the Starr Luxton Lab. And then we were lucky enough to get very generous funding from the Paul G. Allen Frontiers Group and the Paul G. Allen Family Foundation to allow us to do the project that I'm going to tell you about today, to sort of test these hypotheses of what ANK1 might be doing to the biophysical properties of the cell. All right. So let's think about the inside of the cell. And in most textbooks, people draw a cell, you know, like this with organelles, and then there's this vast empty space, which we just call the cytosol. But we know that this is not empty, this space, and that you should really draw the cell like this. The cell is a very crowded place. There's not really empty spaces. In fact, there's this classical model here of the inside of the E. coli cytoplasm. And you can see there are large macromolecular complexes, and it's completely packed in here. There's no empty space. These macromolecular complexes are all bumping into each other. It's a very crowded place. On top of this macromolecular crowding, there's also all the organelles and the cytoskeleton, and you can imagine them creating little compartments or constraints within the cytoplasm. So the cytoplasm is crowded, but then also the cytoplasm is cordoned off into all these different constrained areas by membranes and filaments. And so how do we study just how crowded the cytoplasm is? And traditionally, one approach of this is to use rheology. And rheology is the study of how particles move in a fluid. And the way to do this in cells, traditionally pioneered by the Wirtz lab, was to literally shoot particles, bombard particles with a gun into tissue culture cells. And these particles, these little fluorescent plastic beads of known size and known brightness, then once you have them in a cytoplasm of a cell, you could track them over time. You can watch them diffuse around over time. And you can learn about the biophysical properties of the cytoplasm by the way these particles are tracking around. And the Wirtz lab showed back quite a while ago now in 2008 that if you disrupted KASH proteins with this dominant negative KASH construct, that these beads explored a much larger space, suggesting that the cytoplasm was less crowded or less viscous, even far away from the nucleus. All right, and this was we really didn't think about this a lot for 10 years or so, but we've been thinking about this a lot more now. Now there's some limitations with this approach. You can't really shoot particles into very many systems. You can do this in a flat monolayer tissue culture cells, but you can't do this in a developing organism, in different tissues of a developing organism at different times of development. And this is low throughput and a fairly difficult process to do. So how do we do rheology in a live tissue? And the breakthrough that allowed us to do this work was a few years ago when Liam Holt, who we're now collaborating with to use these tools, Liam Holt at NYU developed what he called genetically encoded macromolecular nanoparticles, which we affectionately call GEMs, as a tool to study crowding and constraint at the mesoscale. And what are these GEMs? These GEMs are an encapsulant scaffold that self-assembles from this archaeobacteria protein. This protein is a scaffold that you can fuse to a green fluorescent protein, and then you can express this open reading frame from any promoter you want in any tissue you want. So it's genetically encoded, and then this scaffold will self-assemble in these 180 mers, and you get these very uniform particles of about 40 nanometers across. And they're very bright because they now have 180 copies of GFP. So we have bright, uniform size, known size particles, and we can now watch these things diffuse around inside the cell to get an idea of the biophysical properties. And these things are on the order of the size of ribosomes. They're a little bit bigger than a ribosome. And it's sort of like a dance party. Early in the evening, the dance floor, there aren't a lot of, it's fairly uncrowded. These things, the GEMs can explore large areas of the dance floor. Later in the evening, when it's very crowded, these GEMs are just going to sort of jiggle in place like a dancer would on a crowded dance floor. And Liam showed very nicely, and others have shown, that ribosomes are one of the main regulators of the crowdedness of the cytoplasm. All right, so we wanted to take this tool and use it in C. elegans to study crowding in tissues. And we got this to work. And Zhang is the grad student in the lab that's been doing a lot of the work that I'm going to show you today. And you can see green dots now diffusing around in the cytoplasm. I think this is an intestinal cell, or no, this is a hypodermis cell. And we've got this, today I'm going to show you in a couple tissues, the hypodermis and the intestine. And then we can track these particles. So here what I'm doing from the intestine or from the hypodermis is we've just sort of put 64 random tracks of random GEMs, and then we can place them all on top of one another into what we call a hairball plot. And you can see these GEMs, many of them, most of them don't move very much this is over a second. We take about 50 frames a second. Most of them aren't moving very much, and a few of them are moving around a little bit. Okay, and then so we can get a feel for how much they're moving. But now we want to start to increase the throughput, and we want to look at thousands of tracks at once. and start to quantify that. So how do we quantify particle tracking? We use a tool called mean squared displacement, which is going to measure the distance that a particle will travel in squared microns over a certain time lag. So this particle here, with a time lag of 1 frame, it keeps migrating, or it keeps diffusing around. You can add take the mean of all these distances with a time lag of 1, and then that mean of the mean squared of the movements here, that's the MSD at lag equal 1. Then you could do this at other times. So like here at time 3, you take a spot from 1 to 4. How far did it move from 1 to 4? How far did it move from 2 to 5, 3 to 6, and so on? Take the average of that. That's the mean squared displacement at time 3. And you can do this at, at all sorts of different times. So here, 1, 2, 3, 4, all the way up to 9. And you keep sampling this, and then you can plot the mean squared displacement over this time lag, and you'll get a curve. And the curve will look something like this. All right. So here we're plotting the mean squared displacement at different time lags. And if the curve is linear, that suggests that these particles are diffusing as predicted in normal diffusion. If the curve is exponential with an an exponent of greater than 1, that suggests that these particles are moving in a directed migration of some sort. There's some sort of flow movement or active movement. And if the alpha is less than 1 and this curve levels off, it's a confined diffusion, and the particle is confined by something else. And so you can measure this. And if you plot this on a linear scale, mean squared displacement over time on a linear scale, the slope here of this linear part is equal to the rate of diffusion, which we'll call the diffusion coefficient, effective diffusion coefficient. Okay. Now, if you plot mean squared displacement over time on a log-log scale, so you have the log of the mean squared displacement over the log of time, now you can see the slope here of these curves will tell you what alpha is. So if the slope, so if alpha equals 1, it's normal diffusion, alpha greater than 1, active diffusion, and confined diffusion. And the intercept here will tell you the diffusion coefficients. All right, so I just sort of wanted to go over that a little bit so you can see where we come up with these two key measurements, the diffusion coefficient and alpha. And this has been done by lots of people in rheology, and I'm taking some data from Liam Holt's paper from the Holt lab. And in mammalian tissue culture and in yeast, these 40 nanometer gems, they diffuse at around 0.3 to 0.5 microns squared per second. And they diffuse in a normal way with an alpha of equal about 1. So I just want you to keep these in mind as we look what's happening in the worms. All right, so here are these numbers again. Mammalian cells about 0.5 with an alpha of 1. These now we have plotted thousands of mean squared displacements from thousands of different tracks of individual gems in our worms, okay? And this is in the intestine. So when we get thousands of gems and then we can average it, the average is way down here because there's lots and lots of lines on top of each other way down here. Okay, then we can look at the slope here and get the diffusion coefficient. The diffusion coefficient here is very slow, about 0.017 microns per second, much, much slower than the diffusion in mammalian cells. Okay, then we can plot them on a log-log scale and we can get an alpha, and we can see alpha, the average alpha is far less than 1, suggesting that these gems are constrained. So they're moving very slowly and they're constrained in place. We get very similar data from a separate tissue. So this isn't a tissue specific thing. If we look at the intestine or the hypodermis, these are in young adults. We get these very slow diffusion rates and these alphas of far less than 1. Okay. This is a little bit hard to look at with all these different mean squared displacements on top of each other. So what we decided to do was to plot from each track, each single gem, we could plot on an XY, we can plot the diffusion coefficient to the alpha, and we can start to learn something more about the population. So this is what we're doing here. Each dot here is a gem, There's a heat map here, so there's a lot of gems here. And what we're plotting is alpha on the y-axis and the diffusion coefficient on the x-axis. And what we see here is that that in either the intestine or the hypodermis, there's two populations of gems. There's a large population here with an alpha of less than 0.5, so very constrained population that's moving incredibly slowly. Okay, and there's a second population here that's less constrained with an alpha around 1 and is moving faster than these gems. All right, and we can pile this up, and I'll show it more like this, where I show a histogram of the diffusion coefficient that we can fit with these Gaussian curves to give us an idea of the size of the two populations and the speeds that these two populations are moving. Instead of taking a bulk average, I'm now showing the average of this population and the average of this population's diffusion coefficients. Okay, so I went through a lot to sort of explain how we saw that there's two different populations. So the conclusions from this part is that these gems in C. elegans are very heterogeneous. Most of them move very little and are constrained in place, with an alpha less than 0.5 compared to very different than mammalian tissue culture cells or yeast. And their diffusion coefficient is very slow, an order of magnitude slower than tissue culture and yeast. So what we take from this is that it's important to look at these properties in vivo, in a living tissue, and to not make all our conclusions from tissue culture cells. And that in a living tissue, these gems behave in an almost glass-like property where they're just moving very, very slowly and they're constrained in place. All right. So what can we use these tools for now? What's the role of ANK1 in this process? How does ANK1 regulate the physical properties? All right, so here's just your eye, and maybe you don't need all the math after you see your eye. So here are gems in wild type, and here are gems in an ANK1 mutant at the same speed, and hopefully you'll see that they're moving around faster, but you probably want to see that in a second tissue. So here it is in an intestine in wild type, they're barely moving around, and in an ANK1 mutant, they're moving around a lot more in the ANK1 mutant. But we want to quantify this. We want to see this. So first we see the hairball plots. In wild type, most particles aren't moving very much, but you can see in ANK1, they're all moving a lot more, and this hairball plot is a lot more of a mess, suggesting there's a lot more movement in an ANK1 mutant. So let's plot each gem's alpha over the diffusion coefficient. And in the ANK1 null, you can see in wild type, most of them are way down here, moving very slowly with a low alpha. In ANK1, the population has shifted to here with an alpha around 1 and a much faster diffusion coefficient. And I think it's easier to look at it this way. So in wild type, you have this population that's not moving very fast. And in ANK1, the population shifts to this one that can move. And even the ones that are moving here are moving at a faster speed than wild type was. Okay, so ANK1 moves the ANK1 mutant causes these constrained gems to become unconstrained, and so we think that ANK1 is constraining the cytoplasm. All right. So what parts of ANK1 are important here? And I didn't show wild type here, but if we delete the KASH domain, it looks like wild type. And if we delete the calponent homology, the active binding domains, it also looks like wild type. However, if we delete the KASH domain plus the transmembrane domain, now we get what looks a lot like the null, where we've lost our constraint. So we think that ANK1 constrains gems by binding to the ER membrane through its transmembrane domain. It doesn't need the luminal KASH domain, just needs to get to the ER. And then the spectrin repeats could act as a scaffold, which most spectrins do. They scaffold membranes to set up some sort of constraining mechanism. Okay. So our summary at this part is that C. elegans tissues are very crowded and constrained, that if you get rid of ANK1, you can get rid of the constraints, and that ANK1, that this role of ANK1 in constraining gem diffusion is independent of the link complex, and actually is a role more generally of the ER. Okay, so what about ribosomes? I told you before that ribosomes are a main regulator of crowding in the cell. And as a geneticist, 20 years ago, I was looking for mutants that caused ANK1-like phenotypes where nuclei were clustered. And the only gene I found in my forward genetic screens other than ANK1 was a small subunit of the ribosome, RPS15. And as a geneticist, I'm like, ooh, ribosomes, I don't want to touch that. And I just didn't think about it for 20 years until we were doing this work, and I realized, hey, there's probably something important here. And so we went back and confirmed this. If you knock down either one of two subunits of the small subunit of the ribosome, RPS15 or RPS18, using RNAi, we get a nuclear anchorage phenotype where we see nuclei touching each other. And here it's quantified. The nuclear anchorage phenotype here is a little bit worse even than the UNC84 phenotype. Okay, so ribosomes are needed for nuclear positioning. What do the gems do in ribosomes? So the gems in here's in the control. They're not moving in the ribosomes. They move. The population moves. Some part of the population moves here, but most of them still in the ribosomes are not moving. Okay, most gems remain not moving, but the gems that do move are now moving faster than in wild type. So wild type it's 0.05, and in the ribosome knockdowns it's 0.09 or 0.08. So they're moving faster. So that's what we expect for a crowding agent, is if you reduce the crowding agent by reducing the number of ribosomes, the gems will diffuse faster. Okay, but they're still constrained by the ANK1 network. Okay, so what about a double mutant? So this is the data I just showed you. In the ribosome mutants, knockdowns, they're still constrained, but the ones that aren't constrained are moving a little faster. And now we started making doubles. So here we make a double mutant, the ANK1 null and RPS 15 or 18. Now most of the gems shift from constrained to unconstrained. So now they're unconstrained and they're moving faster. So the main point I want to make here is that there's an additive phenotype here. ANK1 and ribosomes are working in two different pathways. We think that ANK1 is constraining the movements of macromolecules and that ribosomes are acting as a crowding agent. And when you get rid of both of these, you get the largest increase in gem motions and movements. Okay. What about the ER? Okay, so I showed you the ER, the sloshing ER phenotype at the beginning of the talk. What is the ER? And so here's just a still of the ANK1 ER, and we've used we've drawn a mask around here, and we can just quantify the ER by looking at the percentage of occupancy. Okay, so we've drawn the worm, and then we just look at the percentage that's ER and the percentage that isn't ER. And in ANK1 null, it's about the same as wild type. About 50% of the cytoplasm is ER, even though it looks different. Okay, it's still covering about 50%. All right, maybe a little bit less here. Sorry, it's covering a little bit less than 50%. The ANK1 null. So you see more black here than here. Okay, but the complexity, which is a measurement of how complex the border is, the blue lines here, how complex the border is, it's equally complex in the ANK1 null and wild type. Right. When we look at ribosome knockdowns, you can see just right away there are these large holes in the ER when we knock down the ribosomes. And if we look at occupancy, there's a slight decrease of occupancy, but there's an even more significant decrease in complexity. So we think this is two different phenotypes, and sort of the best way to describe this as two different phenotypes is I showed you before that in an ANK1 null, the ER and all the organelles are sloshing around, they're unanchored and they're fragmented. In a RPS knockdown, ribosome knockdown, the ER is definitely morphological defects, but it's not sloshing. It's still remaining connected to each other and it's moving as a unit. So While they both have ER morphology defects, again, we think it's through different pathways or different mechanisms. Okay. So I just want to spend the last couple slides as an intermediate filament talk, so I thought I better talk about lamins here, at least one intermediate filament. And a couple of years ago, we published this paper in PLOS Genetics, where Ellen in the lab made a bunch of CRISPR mutants. There's only one lamin in C. elegans. And she made a bunch of CRISPR mutants across lamin. And the idea here was that we could use these to model diseases in C. elegans. So the red point mutations are in residues that are associated with cardiac and skeletal muscle defects in human laminopathies. The yellow or tan ones here are cardiac muscle specific, and the blue ones are variants of unknown significance. And she modeled these, and then we put them through a variety of phenotypes. And the first thing we just looked at is fitness. The red phenotypes, the ones that have the strongest laminopathy pathologies in muscles, they have a significant decrease in brood size from wild type to the mutants here. And the ones that are born have a significant increase in lethality. So this one's even 100% lethal compared to wild type. So we sort of recapitulated the severity of the disease pathologies in the worm models, where the more severe pathologies have the more severe brood size and lethality defects. We then looked at a more physiological measurement of swimming. So here are worms thrashing in liquid media, and you can see we have an automatic counting machine to count how fast they're thrashing. And you can see in some lamin mutants, this worm is just not swimming very well. There's severe defects here related to swimming. And we can quantify this all. And the mutants that had the worst swimming defects were also the mutants that cause, that are linked to the severe pathologies in the patients. All right. So we've now got these alleles that are probably reasonable knockdowns of lamins or loss of function of lamins. And we wanted to look at the GEMs and the ER and the nuclear positioning in these alleles. And the first thing here that we've noticed, this is unpublished work now, is that these most severe alleles, these three, most severe ones, have a significant nuclear positioning defect. So this is the worst nuclear positioning defect we've ever seen, except in ANK1. So ANK1 would be up around 60%, but these ones are even worse than the UNC84 nuclear positioning defect. So lamins are also involved in nuclear positioning. And then we looked at the GEMs in lamins. And you can see by the hairball plots that in these 2 point mutants, there's certainly more movements around than there is in wild type. And then you can look at the distributions of the diffusion coefficients of the individual GEMs. And you can see that in the lamin Y59C or R64P, the diffusion moved from 0.59, the gray line here, wild type, to significantly increase in the lamin point mutants. So this looks a lot like the phenotype we saw in the ribosome knockdowns. So let's look at the ER. In the ER, this is a heterozygous control. There's not really there's no phenotype here from wild type, but in the homozygous R64P mutants, you can see the complexity of the ER has gone down compared to wild type, or the balance. This again doesn't look like ANK1. There was no change in this perimeter complexity in ANK1, but it does look like ribosomes. And so if you look at the GEM phenotype, the ER morphology defect, and the nuclear positioning defects, we think that the lamins are regulating macromolecular crowding through the ribosomes and maybe perhaps through the nucleoli. And it's just an interesting thing to think about. I know a lot of people here think about pathology, mechanisms of pathologies of laminopathies, and perhaps macromolecular crowding is contributing to laminopathies and something we can think about moving forward. All right. So the main conclusions here from my talk today is that The cytoplasm in vivo tissues of C. elegans is very heterogeneous, extremely crowded, and constrained. This is quite different and surprising from what others have reported in yeast cells and mammalian tissue culture cells. And then the mechanisms that this is done in C. elegans, one of them is through ANK1, this giant KASH protein, which we think is probably scaffolding the ER in general and constraining the cytoplasm so the gems can't move around too much. In a parallel pathway, we think ribosomes are acting primarily as crowding agents to regulate the diffusion of macromolecular complexes, and that lamins are likely functioning in the ribosome pathway. And where are we going with this next? Well, what we ultimately want to figure out is we want to use gems in C. elegans to understand how the biophysical properties change throughout development, different times in development, all sorts of different tissues. We've now made C. elegans lines with gems in about a dozen different tissues, and we're starting to quantify those. And just, we are seeing differences. So this is a neuron here, and in this neuron, the diffusion coefficient is much faster than it is in the intestine and the hypodermis. So different tissues are going to have very different properties. And then we're starting to look now at how this changes during aging. And hopefully I can show you about that the next time I talk. So I just want to finish with the acknowledgments. As I pointed out along the way, all this unpublished stuff that we did today was done in a very close collaboration together, 50-50 collaboration with Gant Luxton. These are the three folks that did most of the unpublished data I showed you today. Hong Yan's gone on to a postdoc at Yale, and Zhang Yi is the current grad student, and Daniel is our computational specialist. These are other people in the lab working on projects I didn't talk about today. These are alumni in the lab that talked about some of the work I did today. Of course, I want to acknowledge again Liam Holt for getting us started with these gems, and the NIH and the Paul Allen Foundation, without whose support none of this work would be possible. Thank you.